and welcome to this Integrated DNA Technologies webinar on finding the right primers using NCBI for RT primer design. My name is Hans Packer, and I will be serving as moderator for today's presentation. The presentation will be given by Adam Clore, who is a scientific application specialist at Integrated DNA Technologies. He has a background in microbiology and chemistry and studied the molecular genetics of Hyperthermophilic Archaea and their viruses for his PhD in biology. His roles at IDT include assay design and troubleshooting, new product development, research, and scientific writing. Adam's presentation should last about 25 minutes, and we will follow the presentation with Adam answering any questions you have. As attendees, you have been muted, but we encourage you to ask questions or make comments at any time during or after the presentation by typing into the questions box located on the right-hand side of your screen. At the end of the presentation, I will direct your questions to Adam. Also, in case you need to leave early or want to revisit this webinar, we are recording the presentation and we'll make the link to the recording available a few days after the presentation via email. So now let me turn this webinar over to Adam for his presentation entitled Finding the Right Primers Using NCBI for RT-PCR Primer Design.
Thank you, Hans. I'd like to begin today just talking a little bit about what we do and sort of the process that we go through when we're designing RT-PCR primers. So there's several different considerations during this design, and I think most people are familiar with the considerations that surround the physical properties of the primers and probes. That is the melting temperature, the presence or absence of secondary structures such as hairpins, and things like that. But one of the things, actually a couple of the things that are not often considered when designing real-time PCR assays are the presence or absence of transcript variants and the presence or absence of SNPs, or single nucleotide polymorphisms, within the primer and probe area. And that's what I want to focus on today, is understanding SNP variants, understanding transcript variants and the locations of SNPs. And I'm going to go through how we can figure these things out, mainly using the tools that are provided by NCBI. So to begin with, what is a transcript variant, and why should you care? In most eukaryotic organisms, genes are expressed, and the immature mRNA contains both exons and introns. During mRNA maturation, the introns are cleaved out, leaving only the exons, and these exons are what is then exported into the cytoplasm and is eventually coded into proteins. One of the things that has been found in recent studies is that a lot of exon splicing happens under is a variable. These things can be changed depending on the needs of the cell. And so if you look at different genes, there are many alternatively spliced transcript variants that are found. In fact, about 95% of the human genes have transcript variants in them. And so what these transcript variants are basically different combinations of exons being present. So as you can see here in this gene where we have 4 different exons, you can have a transcript variant that contains all of the exons or just a subset of the exons, and these make up the variants that are present in there. This is what happens often in the cell. What happens to a lesser degree, but is definitely not unheard of, is splicing a part of a exon. So for instance, we could have a gene that has all 4 exons, and we could also have a gene that has all 4 exons but is perhaps missing the first few bases of one of the exons. So that's another type of variant that can appear. The roles of the variants are really varied. They're in normal gene expression, they're used, transcript variants can be used for anything from gene regulation to actually producing different products, and in some cases are the result of aberrant splicing. But just to give an example of one of probably the most well-studied transcript variants, I'll talk about the double-sex gene. This is a gene that's present in Drosophila, and it influences sexual development. The gene has 3 exons in it. The pre-mRNA has 3 exons, I should say. And these exons are spliced in a way to either create a female variant or a male variant. And depending on how this is spliced, the sexual development of the brain and the body will develop under different conditions, or they'll develop differently. So there's a female variant and a male variant. And regardless of the actual sex of the organism, this splice variant which splice variant is present will decide the male or female body type that will be built by this insect. As I mentioned, transcript variants can also result from aberrant splicing, and this often leads to disease or is at least associated with diseases. And this is just a very short list of a few of genes that contain splice variants that lead to one type of disease, cancer. This is just, again, a very short list, and there are many other splice variants that are associated with many other diseases. So I want to go through now and discuss how we can identify transcript variants and sort of what those transcript variants are doing when we have a gene of interest. And as I mentioned before, I'm going to use the NCBI database for this. So to start out with, if we just go to the main page of the NCBI database, we can enter in a gene, and in this case I've chose to enter in the the PKM2 gene. I'm going to go ahead and hit search on this, and that will bring up the entrees page, which will show a variety of different databases here and the hits within those databases. What we're going to be interested in is looking at the gene database. As you can see, there's 61 hits on this. And when we click on that, it brings up the results of those hits. And as you can see, there's a PKM2 gene in a variety of organisms, human, mouse, rat, so on and so forth. We'll go ahead for this example and select the human. And that brings up a page that contains a lot of gene information, and included in that gene information is a graphical representation of the transcripts of that gene. You can see here that this gene has 7 different transcripts, and in this graphical representation you can see the location of the exons, which are noted by these vertical lines, and the introns, which are noted by the horizontal lines here. And you can see in each of these variants there are different exons, presence and absence are different in these things. And so we can get an idea of what these splice variants look like. One other thing to note, you can see that there's a light green section and a dark green section. The light green section indicates where the untranslated regions are located, and the dark green section indicates the protein coding regions of this particular transcript. If we want more information, we can go ahead and select this hyperlink up here, which goes to the reference sequence details. This is information that's located lower in the page. That information gives us some more specific information about the individual splice variants. You can see by the refseq number, or the refseq accession number here, beginning with NM, that these are messenger RNAs. And so if we look at the different NM accession numbers, we can get an idea of what type of variant this is by the description that's located here. We can identify which one's the longest. There's usually some information about which particular exons are missing in the cases where they're missing, and some other information about that. Along with that, there's often some information indicating the presence of or correlation with, for instance, a type of disease or just some general information like that. So it's a good place to just generally start to get an idea of what the different transcripts are and what these different transcripts are doing. Now, once we have an idea of what the transcripts are, and once we have an idea of what they're doing, you can make a decision about whether you want to target all of the possible transcripts in a gene or just single transcripts in a gene. And once you've made that decision, you can go to our design tools that are located at the Integrated DNA Technology website. These are tools that are all freely available. There's no login required. And you can go ahead and use these tools to design your assays. There's 2 different tools that are going to work well for this, and these 2 different tools will work slightly better for different conditions. If you are using a human, a mouse, or a rat model, The best tool that we have is the Realtime PreDesigned Library. This will be a library of pre-designed assays for all of the genes that are in the RefSeq database. These designs have all been screened for crosstalk between different variants, and they've also been screened for the presence of similar sequences located elsewhere in the genome, so you can be assured that the assay is only going to target that particular area and not some non-specific area. So these pre-screened assays have a great deal of screening that has went into them. What we offer for individuals that are not working in human, mouse, or rat is a real-time design tool. This tool will allow you to enter in RefSeq numbers or just sequences from any organism. And by doing that, you can design specifically for that that sequence that's in there. The downside to using that is you don't have the ability to screen across the entire genome and make sure that there's not problems elsewhere. We'll go into that in a little more detail in a minute here, but first I want to go into the Realtime PreDesign and give you guys an idea of what that looks like. So going to the IDT website, you can access the Realtime PreDesign Library. It's under, you can find it either under Products or under SciTools. Once you get to the PreDesign Library, you can enter in gene information in several different fields. You can either enter in the gene symbol, as I've done here, you can enter in a RefSeq number, or you can enter in an assay ID number. This assay ID number is IDT's specific number for the assays that we've designed. You can also then select human and mouse and rat, or just one of the few. I've went ahead and just selected human, and just like before, I've entered in the PKM2 gene, and this is showing the results of that. So you can see that there are several different assays that are designed here, and they're targeting different locations, as you can see by the exon. One other thing to note is it indicates here whether you can detect all of the variants, in the cases of several of these, or not all of the variants. Now, if we want to know, in the case of these ones that don't select all of the variants, we can mouse over and see exactly which variants this is targeting. And as you can see in this this particular example here, there are 4 variants that are targeted. These are the accession numbers of the 4 variants that are targeted. One other thing to note with this, if you look carefully, you can see that the exon location here is indicated as exons 13 to 14. Most of our assays are designed to span exon junctions so that you eliminate the possibility of amplifying genomic DNA from this. You can see on these transcripts here that the exon location is different. And I want to explain this difference in exon location. This comes from the fact that for quite a long time, NCBI did not have an official convention for exon numbering. So often it was done in a manner like this, where in this example here, there's 2 transcript variants. They're missing different exons. And the exons are numbered simply by the order that they appear in each of the variants. So what you can see is this results in some confusion when you're comparing variants, because in this example here, exon 4 in variant 1 is the same exon as exon 5 in variant 2. One of the ways to get around this, and kind of the way that NCBI is moving now, is to using a numbering system that is based on the gene itself and not on the variants. So in this case, the exons would be numbered from 1 to 8, regardless of whether or not they're present in each of the variants. That means that similar exons have similar numbers, but different variants may be missing exon numbers. In the case of IDT, what we have done is we have kept this type of numbering system for the external view. That is this view here. So when you're looking at, when you're comparing all of the different assays together, you're seeing exons numbered from the genomic context. But in keeping with the numbering system that's present in many of the the individual variants in the accession files, we've kept the numbering system for the individual variants to be the numbering of exons within that particular variant. What that means in this case is that there's 13 or 14 exons that are present in the genome here when you get to this location, the location of this assay. However, these variants here are missing a few of the exons before this, and so their numbering system is a little bit smaller. Their numbering is a smaller number there. Okay. I want to go ahead then and jump into our real-time PCR assay and describe how, or our real-time PCR design tool, and describe how we design assays for that. In this particular tool, you can enter in either an accession number, or you can just enter in a FASTA format of sequence here. Now, if you enter in just a FASTA format of sequence, there's not going to be any information indicating where exon boundaries are. One of the things that you can do to indicate where the exon boundaries are, if you know where they are, is to use a lowercase letter for the to indicate an intron. So, for instance, you could put a lowercase t at the exon boundary to indicate to the system that that is where an exon is located. Because it's in lowercase, the computer system will recognize that as an intron and will ignore that during the design. Okay, so in this case I've entered in an accession number, and when I hit enter, it brings up the next screen here. This screen, because I've entered in an accession number, has pulled from the NCBI database the exon locations, the exon boundaries, so it can tell where the exons are, and I can select here where I want to design my assay. So for instance, if I would like to design my assay to be in exon 1 and exon 2, I can simply select the exons that are included in there. Now in this particular gene, exon 1 is completely untranslated, and part of exon 2 is also in the 5' untranslated region. Part of exon 2 is also a coding sequence, and so the selection here allows you to select the whole exons as I've done here, or if I wanted to, I could also just select the untranslated region by deselecting this option here. I'll go ahead then and hit Design Assays. And what this does is it gives me the list of possible recommended primer and probe sets that can be designed. Now this is a fairly small region, and so it only designed two different it was only able to find two different acceptable primer and probe sets, and it displays the location of those here and the sequence that those are designed for. Okay, the next thing I want to talk about is SNPs. And first of all, what is a SNP and why do I care? SNPs are single nucleotide polymorphisms, and that indicates that there is a single letter change in the genetic code. There is a large number of SNPs that are present in the human genome, and the recent large-scale sequencing that we're doing now of the human genome is just increasing the number of SNPs that we're finding by a very rapid rate. So right now we've identified around 56 million SNPs in the human genome, and of those 56 million, about 16 million are located either in genes, in gene exons or in gene introns. So these are located in areas that could possibly be affecting our design, particularly those that are located in the exons. Most of these SNPs cause silent mutations, meaning that they do not cause a change in the amino acid code, but some do. Regardless of whether they're silent mutations or active mutations, they can be clinically relevant, and this is just an indication, an example of an individual with xeroderma pigmentosum. This is caused by a single nucleotide polymorphism in a DNA repair gene, and it causes individuals to be exquisitely sensitive to UV radiation. So again, if we want to identify where SNPs are located, the best tool that we have is going into the NCBI database, and there's a particular SNP database that we can use to find the location of SNPs. So again, if we go back to the PKM2 gene, which we looked at earlier, we can go ahead and hit the zoom in button, and you can zoom in to the level of where you can see the location of SNPs. Now these locations of SNPs are within an intron region here, and you can highlight over the SNPs, and you can get an idea of the location of the SNP, what the change in the SNP is, and some general information. Now if we want more specific information, we can actually go over to the sidebar over here, and we can click on SNP Gene View. Clicking on that brings up a list of all of the annotated SNPs that are present in there, and also gives some indication of what these SNPs do. So for instance, there's information on whether or not this is going to be a SNP that causes a change in the amino acid sequence. The ones that are causing a change are indicated in red, and the ones that are not causing a change are indicated in green. But the most important thing, I think, as far as designing an assay, is you get an indication of where the SNP is located at. So this is the location in the mRNA where this SNP is located at. Now, if we jump back to the real-time PCR, this is just an example of a design that was done for the real-time PCR. We can click on the button up here and go to the report. And what this report does is it gives an indication of the location of each of the primer and probes. And so this allows us to see where the primer and probes location are and compare that to this table here in the SNP database. And this will indicate, in doing this, we can find primer and probe sets that do not have a SNP located in them. As you can see in this column here, it's the location of the primers and probes. Okay, so just to summarize this, the RT-PCR design tools that we have available are the Primetime PreDesign PCR tool. This is only available for human, mouse, or rat, but the benefit of this is it identifies transcript variants, and it is specifically designed to avoid SNPs in all of our pre-designed assays. So you can be assured that these designs are going to target exactly what you're looking for. Of course, the downside is that these are only available for human, mouse, or rat. So if you're working outside of human, mouse, or rat, the best option that we have available is to use the real-time design tool. This tool will allow you to design very good assays, but it will be up to you to check for the correct transcript variants and the correct SNPs. And to do that, you can go through and use the NCBI tool. All right, with that, I would like to thank everyone for attending the presentation, and we will take some questions.
Okay, I want to thank Adam for a very useful presentation. And Adam will now answer your questions. So if you have a question and have not done so already, please type it into the questions box located on the right-hand side of your screen. Okay, Adam, there are already a few questions for you. Here is the first one. Are the assays that we have, that IDT has, the same concentration as ABI TACMan?
That's a good question. Our assays actually have the flexibility of coming in different ratios of primer to probe. So we can adjust the ratio of primers to probe in the assay and set that up to whatever the customer wants. Now, I'm not sure right offhand exactly what the concentration of the TACMan assays are, but because we have the flexibility to adjust the ratios, we can create an assay that will be similar to that found in the TACMan assays.
Okay. The next question is, do you provide custom real-time qPCR assays with VIC?
Yes, that's a good question too. We don't provide assays with VIC, unfortunately. VIC is a proprietary dye that we're not allowed to use. We do have some alternatives to VIC that are available, and if you look on our our modifications page, we have a list of fluorescent dyes that will emit at the same wavelength as Vic. Now, there's a word of caution that I will give to everyone in switching from one dye to another dye, and that is, in most machines, there is a recalibration that needs to be done, and that's because the overlap of fluorescent signals, even with dyes that emit at the same wavelength, are often a little bit different, and so you usually need to recalibrate your machine every time you switch dyes.
Okay, the next question is, do the primer design tools provide delta G values for hairpins, etc., and does it check for non-specific primers like the Primer Blast tool?
Yes, and that's a good question. The primer design tools do check for delta G values. And you can see up here, we don't have an actual delta G value listed here. I don't believe that this is an exact delta G value, but it does provide an indication of how good. Actually, no, I'm sorry, I take that back. These are delta G values. So this does show the delta G values for each of the each of the probes, and then the probe dimer combination. So probe and primer dimer combination. So if we look at this, there's different combinations of primers that are here, and the dimers that are gonna form from those. So, and I'm sorry, Hans, what was the first part of that question?
So that was the first part about delta G values, and then the second part was, does it check for non-specific products like the Primer Blast tool?
Yes, and that's another good question. There's a difference in our two tools here, and if you're using the Primetime PreDesign Library, this will check across the entire genome for specificity. So it will look across the entire genome and make sure that there is not similar primer binding sites elsewhere in the genome that could amplify. The Realtime design tool only uses the information that you provide it. So it just looks across the particular gene or the particular sequence that you've entered into the design tool. So it's not checking across the entire genome.
Okay, the next question is, does the software have probe selection options?
Yes, and that's a very good question. The software does have probe selection options, and if we just jump back to the picture here, over in this area here, we can actually select on the this unfortunately isn't live, but if we would select on this, it would allow us to open up a window that would show the different options that are available for probes. So that the different options that are available are, there's different synthesis scales, there's different fluorophores and quenchers that can be used, and as I mentioned earlier, there's different ratios of primers to probes that can be ordered as well. So there's several different options with that. With the Realtime design tool, there is a similar set of options that are available. So in both cases, there's a good deal of flexibility with what can be ordered.
Okay, the next question is it possible to design primers for repetitive elements?
Yes, that's a question that we get occasionally, and the answer is variable. It really depends on the repetitive elements. Often if you can find a anchoring site, an area outside of the repetitive element that you can design at least one of the primers for, we can come up with an assay that will work for that. This is a more complex problem, and this is the type of problem that I recommend most customers contact our technical support about, and we can go through and help you design a custom assay for these types of problems.
Okay, next question. Does the qPCR primer blast with the whole genome?
Okay, so this, I believe, is a similar question to the one earlier. And yes, when we blast in the Primetime PreDesign Library, we are comparing the designs across the entire genome and making sure that there is not a repetitive site there that would allow amplification in some area other than the area that we're targeting the assay for. Again, in the real-time design tool, it's only looking at the area that it's only looking at the sequence that the user has entered into the design tool.
Okay, here's another question. How big would the difference be in the result if the SNP that's found is in the 5' region of the primers?
Yeah, and that's a good question. So if we're talking about SNPs being present in primers, the 3' end of the primer is the worst spot for a SNP because that's going to destabilize the end where the polymerase is going to extend from the primer. Now, if the primer is in the 5' end, it may not be that bad. In fact, it may be tolerable. The problem is determining exactly what type of influence this is going to have on your assay. And the best way to test that is to have a template that is both SNPs, both alleles of the SNP, and confirm that the amplification of the qPCR is of identical efficiencies. Now, if one of the SNPs causes the efficiency to drop, you're going to know that there's a bias towards or a bias against that particular SNP. And so a good way to test that, is to use templates from both SNPs. One of the popular ways people get that template, if they don't have examples of that template present in their lab, is they can order a mini gene from us. That's a small gene, usually 400 bases or shorter, that's inserted into a plasmid. We can make those relatively cheaply and relatively rapidly, and they work very nice for template controls.
And here's a related question for you. It says, I have not been screening my primers to check whether they amplify a SNP area. How will this have affected my results?
Yeah, and it's a good question, and the answer is really variable. Part of it depends on what you're using for template. If you're using cell cultures, your cell cultures are likely to be one consistent allele of that particular SNP, and so it's probably not going to have a large effect. Now, if you're using something like clinical samples where the template may have multiple alleles in there, there may be an effect. And again, the only way to really understand what that effect is to take template of 2 known alleles and amplify those side by side and make sure that the amplification efficiencies are similar.
Okay, next question. Does IDT provide microRNA primers or a tool for designing for microRNAs?
Yeah, that's a good question too. And unfortunately right now we do not have a tool that is set up to design primers for microRNAs. Again, that's something you can contact our customer support group about, and you can work with some of our technical specialists to help design those. But currently we don't have any tools online to assist customers in that respect.
Okay, next question is, in your opinion, should I run a standard curve in every run to validate the results?
Yeah, that really comes down to a judgment call about whether you should run a standard curve in every experiment. If you are very confident that your experiment is very reproducible, you can probably get away with not using a standard curve in every experiment. The question is going to come up, though, if you have questionable data or you have variable results and you haven't ran a standard curve in that particular experiment. The question is always possible is that perhaps your PCR has lost efficiency somehow. Perhaps concentrations of something has changed or something like that has happened. And so without having that standard curve in there, if something goes wrong, there's always going to be that question. The researcher really has to think about, if this happens, is it going to be a better use of their time to repeat the experiment or a better use of their time to just spend a little more time and run the standard curve in each experiment? And that's, I think, something that needs to be decided by the researcher.
Okay, here's a question about the PrimeTime software. In the PrimeTime view, how do you navigate to the results to see the actual primer locations and actual sequence data?
Yeah, that's a good question. I actually showed that in here, but let me just show that again because that's a very important piece of this. When you get the results come out like this, and you see the sequence of the primers, and you get a vague idea of where the location of the probes and primers are. But if you want to see the exact location, you go ahead and click on this button up here labeled Report. And let me jump to the other example. So if we click on that report example here, that brings us to this page. And this page here is the results of it's the same information here, but there is a lot more information included in that. So we have the amplicon lengths, we have the locations in the mature RNA, we have the locations in the gene itself, and then also TMGC, hairpin, different analysis like that.
Okay, the next question is, can you explain the IDT Xen probes?
Yes, that's a very good question. The Xen probes are a new type of probe that we're offering. It's very similar to a dual-labeled probe or a TACMAN probe or hydrolysis probe. All those are basically describing the same thing. The difference is with the Xen probe is that we have not just a quencher on the 3' end, but we have both a quencher on the 3' end and an internal quencher. We do this for a couple of reasons. One of the reasons is when you get really long probes, these are generally probes that are designed in AT-rich areas, the quencher gets far enough away from the fluorophore that the quencher doesn't quench efficiently. And so by putting an internal quencher in there, we can even with long probes keep a quencher close to the fluorophore and keep that fluorophore quenched when it's not before it's been digested. The other benefit of it is just simply having two quenchers on there seems to cut the background down to a lot lower number. So what you end up with, even with both short and long probes, is a very low background probe, something that is going to start very low and allow you to see a very large increase in signal once the hydrolysis happens.
And a follow-up question to that is, are there any worries about steric hindrance with Xen?
Yep, and that's a great question too. It's something that we've studied quite a lot in our in-house research, and we don't find a steric hindrance to the binding. So a lot of people are worried that this internal probe is going to disrupt the binding. What we find is actually the exact opposite. We find a very slight increase in the stability of the probe. It's an increase that is only about 1/2 degree or so, but there's a very slight increase there. So it doesn't seem to be creating a steric hindrance. If anything, it seems to enhance the binding affinity of the probe, but only very slightly.
And another question here. This one looks like it's talking about the variability with replicates. So what cutoff value of CT standard deviation would you recommend for replicate RT-PCR with IDT primers?
Well, I don't think that this is a question that should be specific to IDT primers. I think this is a question that is really specific to the goals of the researcher. Generally, the deviation that you're going to have in your replicates is going to control how accurate of a prediction you can make on the quantification of the material you have. So if you need a very accurate, if you need to predict the quantity of material you have very accurately, you need to use CT values. You need to have a standard that has very tight CT values. To a certain extent, you can increase that by increasing the number of replicates that you're using. But it may be that you also need to go back and work with the design, with pipetting and with different issues like that to increase the repeatability of the assay. But ultimately, this comes down to the precision that is needed to answer the question in the experiment.
Okay, and I think that that is all the time that we have for questions. I want to thank you all for attending Adam's presentation today. This is one of a series of webinars that IDT will be presenting on qPCR, as well as other topics. We will email you about these future webinars as they are scheduled. We also would like to send you the link to the recording of this presentation in the next day or two. Of course, you can always just go to the IDT website and find it under the Support tab. Thank you again, and best of success in your research.